Hello.
Hi.
How are you doing?
We had an exciting SLAM meeting.
Oh, really?
Yeah, on Monday. He was doing starfish oocytes, and they're wild type starfish oocytes, and he was looking at the wave that goes across the oocyte, fertilization wave, which is sort of like a differentiation wave. And so it was, he's been studying these, and I actually copied his lecture, but it's online if you wanted to see it. So they were exchanging emails about it because of the axolotl waves.
Yeah.
Yeah, who was it? Here, just a second. Peter Foster.
All right.
Yeah. So Peter Foster, Brandeis University.
Okay. Yeah.
Yeah. So that's who's doing that work, and it would be really great if he would look for other waves in multicellular starfish oocytes.
Yeah. Yeah, that would be good.
And like I said, I thought I saw an equivalent wave in the zebrafish, or some sort of thing that you would consider a wave, because somebody was studying how the notochord was formed, or I don't know, sort of the Henson node that moves. These zebrafish have their sort of equivalent. And you had a paper on it, and I, at one point, while reading it, I got confused as to what they were talking about, because they had encapsulated cilia, and they were turning.
Right.
And rotating, and that broke the left-right symmetry. And then I'm saying, well, so what, it's turning inside this capsule, how does that affect the outside of it? Like that, they hadn't connected all the dots, I guess.
Yeah.
They were probably right, but it just like I wasn't getting where the forces were going by reading it. So, yeah.
Yeah. Well, that's good. So that's online, the Slam Talk?
Pardon?
The Slam Talk is online?
Yeah, it's online.
Okay.
Yeah. Peter Foster Slam Talk on Monday.
Okay. Yeah.
So I guess that's the 18th. It was on, and it was a good one.
Oh, thanks. Yeah, so, that sounds good.
Yeah, it was recorded.
Yeah, yeah. All right. Well, thanks for that update.
And he's looking for PhD students. Like, he's just a new researcher, and he's opening his own lab. And so if you're willing to go with untried new researcher, he's there.
Yeah.
And so that's an opportunity for people.
Yeah.
Pardon me.
That's okay.
I'll turn this off my voice.
All right. Well, thanks for that update. So Harikrishna and Karan, hello, welcome.
Hello.
Hi. So yeah, I wanted to have a meeting this week because we had a Miss Monday. I was at a workshop, so I couldn't make the meeting time, but I wanted to get some updates on GSoC related things. So who wants to go first?
Should I start?
Yeah, go ahead.
Yeah. Okay. Hi, Bradley. Hi, Susan.
Hey.
Hi. Yeah, this week, from 18 to 24, I have to try out different projection methods. So that is going on. Otherwise, previous week was, I think, mostly covered, in the week 4 update where, I was trying out different angles for that, for transposing the contours, on a fixed axis. So the angles, projection methods are somewhat similar, like they're not done in much changes, like to the way of, finding the angle for the contour and, how much I should rotate it before putting it on the axis for the 3D model. So that is there. Otherwise, this week's thing, coming out with different projection methods for generating, for projecting it onto the model. I think equirectangular projections that we had discussed, weeks ago. I'll probably be going ahead with that only, but I'll still keep, looking for ways of tweaking the projection method that is there. Otherwise, the equirectangular projection system is the most promising right now, because it has the. I think it'll be more accurate when it comes down to plotting those 3D coordinates to that particular image that is there.
Okay, that's good. What are you going to do this coming week? What are your plans?
Yeah, so this is the one where I'll have to take the cropped out image of the embryo, and I'll be using that to generate a projection image, in which I'll transpose it onto an equirectangular projection image format, where it'll kind of be like when we look at the Earth, when we look at the Earth on a 2D map, it's something like that. So I'll be applying a transformation where I'll be converting it from that to the equirectangular projection image, and then I'll be using that equirectangular projection image to transpose it onto the 3D model. So this is where I was supposed to try out till I think July 24th different ways of going about this projection method part. So I'm still looking forward. I'll still keep looking, for different methods for doing that. Otherwise, the equirectangular projection formula, the transformation formula for doing that is the one that I'll be applying.
Okay, that sounds good. Thank you for that update. And you're not running into any problems, technical problems or anything?
So far, no. I keep on trying out new things. So most of them, I tried them partially before also. So I was thinking of making them work, but I think I either have to tweak the current existing method that I've laid out to generate better approximation at each step rather than trying out a different thing. I think that would give me, I think, a better result, because like with the previous set, like I was generating outlines on my own. So initially I just had 8 outlines. So using averages, I'm generating more outlines on its own. So I think I'll have to another thing that I've not, I think, included right now is generating a metric, for comparing how much more am I approximating rather than the real thing. So, maybe a metric to decide the percentage of deviation, maybe something along those lines. The error percentage rate, maybe something along those lines. Otherwise, I think, yeah.
Yeah, that sounds.
Otherwise, things are going on okay.
Okay, that sounds good. Sounds good. Harikrishna, how are you?
Just a minute.
Okay.
There's some background noise. Yeah, hello. Yeah, so this week I tried something new. What I did was, from the embryo images, from the different images, I will find out what I will basically what I'm doing is not projecting on a sphere, but something of a different shape, like with three different radiuses, the X, Y, and Z radius. So those radiuses can be found out from the images, assuming that taking a convention like diameter is 2 millimeter of the embryo. So from that, I'll be taking out three radiuses from radii from the images, from the data sets which I have, and creating the 3D coordinates of the vertices of the 3D model. So from those vertices, then I can render the triangles. Like the three vertices can create a triangle and create a overall mesh. So that would be the 3D mesh of the embryo. And now, right now, I'm trying to learn a bit about UV mapping, so that I could project those images onto that mesh. So I think UV coordinates can be helpful for mapping those images onto the 3D mesh. So that's what I've done this week.
All right, sounds good. Have you had any technical issues that you need to address?
On the the process of for the process of creating the 3D model, I think there's no technical issues till now. I'm still exploring the ways to project the image. Okay. I had some issues with the UI part, which I was making using the JavaScript libraries and frameworks, but I have solved it. Now.
Okay,
so right now there are no issues.
Okay, that's great. So what are your plans for this coming week?
I want to learn more about, discover more about UV mapping, and try projecting the embryo images on the 3D mesh which I have created. But also I'll be trying to do something with the UI also. I want to build it for UI.
All right. Well, that's good. Thank you for that update.
Yeah.
Yeah. So I don't think Jiahong or Wataru can join us today, but we've been communicating in Slack about some of their work, and they've been working in their own repo on a lot of this stuff. So I don't know if I can really explain this well because I'm not in the repo. It's not actually my repo. So let me go into their location here. I think it's this one. Yeah, he sent me a link to. Oh, this is a different one. Okay, so they have a repository here they call DevoGraph. Here it is. So this is DevoGraph. This is where Jiahong and Wataru, and they have someone else working with them on it. It's not a GSoC student, but they're going to work on it with them. And this is so basically they're pushing to this repo, and then they'll push to the DevoLearn repo. So they're going to incorporate this into DevoLearn, and this is going to be like the graph neural networks component of it. So we have a couple of things already taking shape. We have they divided their project into like stage 1, stage 2, and stage 3. Stage 1 is the image processing. So this is where they're taking images from different databases and they're bringing them into a pipeline. We talked about that last week, and some of the things that need to be like in place for that, some of the checks that need to happen, some of the quality control. But this is I don't know if I can render this notebook in here. But this is stage 2, and so so good. They've prepared a notebook here. They're doing experiments in stage 2. So the second stage is to convert a CSV file into a temporal and directed graph. So this is they gave an update to me a couple weeks ago on this now, and this was part of this idea of taking the image data, segmenting it, getting numbers out of it, and then converting it into these temporal and directed graphs. So basically, they're graph structures. You use the centroids of each cell, and you build these graph embeddings or these graph representations, and you use well, the CSV file has the numeric values of the centroids in the cell volumes and things like that. And then these graphs work from that data, and you use different criterion to build the graphs. So this is a sample CSV file that they provided. Let's see if we can look at it. So I guess I can view the raw. So there's a way to render CSV files in GitHub, but then that's not what we have here. But anyways, so this is just I think the input data from there. I think they're just using raw input data from our repositories for this example. But for a segmented example, you would basically have the XYZ coordinates. You might have the cell volume. You might have definitely the cell ID. So, that'll all be sort of part of that input. And let's go back here. All right, so this is back to the notebook here. And then they're kind of working through, then they pre-process the sample data. So they have this data is formatted here. So then this is the code for that. And they have the lineage data. And they build a DGL graph based on the given sample data. And so this is where a temporal graph is based on the CSV data. Given lineage tree information, we also provide an API that connects mother and daughter cells in successive temporal graphs and generates a directed graph. So these are things that I don't know how far along these are in terms of implementation, but this is the idea. And then finally, okay, so then this is actually Jia Hong's part, I think. So it is, a lot of it is sort of this first draft. So he's got a lot of this laid out. Then finally, the visualization in 3D plots. So this is a three-dimensional plot where he's tracking the path of the cells. So the idea is that they migrate in a, when you capture the data for a single cell, it's going to change its position or migrate a bit, and then it divides, and then you have daughter cells, and they migrate and they divide. And so these are the cell tracks for that. So this is the cell tracking the nuclei over time. It doesn't really say which cells these are, but these are mother and daughter cells. So I think they're actually like, I don't really know. You'd have to explain this to me a little bit more, but there you basically have the cells that move, migrate. The position migrates, and then so it may not actually be the cell. It may be the nucleus or the centroid of the cell. We call it the centroid because we don't want to make any assumptions about the nucleus, because it's not. It's a marker that's expressed in the nucleus, but we don't know exactly. It's not like the center of the nucleus. So, but anyways, it's a good proxy, I think, for movement in the cell and positioning. And so then you can also look at the daughter cells relative to the mother cell, and you can see how they've changed their position in the cell or in the embryo. So you have these types of plots. And then this is this is an example of where you have the mother cells and the daughter cells. So these green dotted lines go to the daughter cells from the mother cell. So that's what stage 2 looks like. Stage 1 is the data. And then I think there's maybe, and then this is stage 3. So I don't really know. We didn't really discuss stage 3 too much, just a little bit. And then this is just kind of moving it into a pipeline and mapping it to some models. And I think that's going to take shape later this project period. So that's what they're doing. So I was kind of hoping that they'd come in, but I think it's pretty late for them. So, but that's what they're doing. Anyone have any questions about that, or?
Yeah, it seems pretty interesting. I think they were using the DGL library, for mapping the embeddings properly.
Yeah. I don't know what the best method for that is. It's kind of like they're flying blind on that too, because I don't know. Like graph neural networks are kind of a new thing, so
yeah.
yeah. But we're gonna
But it seems
experiment.
really, like the way they're trying it, I think it seems like I've not seen it done like that. But
Yeah.
otherwise, it's still pretty, it's like uncertain ground, when you're dealing with GNNs. So yeah, at least they've got the embedding there, so that's pretty interesting.
Yeah, yeah. And the embeddings, because we're integrating it into DevoLearn, we're trying to make it so that people can use it concurrently with segmentation. So we have these pre-trained models that you can use to segment your images, and then you plug it into this, and you can create graph embeddings, and then the graph embeddings can move on. You can use those in different ways. So you can use them to, as data analysis things. You can actually generate a number of candidate embeddings and choose from the best one, for different criterion. So we don't really know, we haven't settled on any one criterion, because you might want to create a graph with different criterion. You might want to create a graph, for example, of like a distance in the embryo. You might want to create one based on some signaling molecule you have a hypothesis about. So that would be like the advantage of having a lot of different embeddings generated, and then you can use it in other models as well.
Okay.
Yeah.
Has everybody got access to the 300 or more images that I posted on Sync? Or oh no, sorry.
Yes, I think.
Yeah, we have access to the Sync.
We have access. Yeah.
Okay, thank you. I just have a very noisy dog this morning.
Okay. All right. Yeah, that's good. Thanks for the updates. I'm glad that everyone's coming along, and we have the first evaluations coming up soon, but those are, I think, going to be pretty standard. I don't think there shouldn't be any problem with that. So they'll be sending along like some feedback for you to fill out in like a week or two. I have to fill it as a mentor. I have to fill out the evaluations, but I think we're all doing pretty well here. So, okay. So I think that's it for today. I wanted to give an update on, or I wanted to get updates on the different projects where we stand, look things over, and get a sense of what needs to be done next. And it looks like everyone's on track for the first evaluation, but also just kind of like no major problems coming up. Okay, anyone else have anything they want to mention before we go?
I would like to put stripes on a ball bearing for some of this work. My eye got operated on. You probably can't see that from my image, but anyway, so I've not been doing that. I have one glass in my glasses, and I see blurred out of here. It's more of a distance thing, so I can try to put stripes on it now that it's sort of on the way to being healed, but it's sort of I don't have good vision at the moment.
Yeah.
If you're wondering where the stripes went, they're being delayed. Anyway.
Wow.
Yeah. My eye's good. They checked it. It's good. So.
Good.
Yeah.
All right. Now I'm going to talk about a few papers in our reading queue. I'm going to cover two different topics today, so let's get started. I'll share my screen. The first topic is light exposure in the embryo. And so we've talked about embryonic behaviors before. It generally proceeds by some sort of muscle twitching or some sort of reactive response to an environmental stimulus, or to the connectome and the muscle being wired up and getting this electrical activity for the first time. And so sometimes it's very non-goal directed behavior. But we actually have some examples of early and late light stimulation and some other early light stimulation work in chicks and some other model organisms that draw out some of these distinctions a bit more. So some of these behaviors are goal-directed, some are not goal-directed, and we'll see a little bit more about this. So this paper is from the journal Symmetry, and this article is entitled Early and Late Light Embryonic Stimulation Modulates Similarly Chicks' Ability to Filter Out Distractors. So chicks from the domesticated chicken, Gallus gallus, learn to run from a starting box to a target located at the end of a runway. So this is a goal-directed test of this running behavior. At test, colorful and bright distractors are placed just outside the starting box. Dark incubated chicks, which is defined as chicks that are maintained in darkness from fertilization to hatching, stop significantly more often, assessing more the left side distractor than chicks hatched after late, defined as for 42 hours during the last 3 days before hatching, or early for 42 days after fertilization exposure to light. So these are, what this is where they have chicks that have hatched and now they're behaving in the world, but they were reared in different conditions. So the first set of conditions involve rearing and hatching, or rearing in darkness, from fertilization to hatching. So it's complete darkness throughout the developmental period, the embryogenetic period. And then the other conditions are chicks that are exposed in different ways to light. So this is for 42 hours after fertilization. This is for 42 hours during the last 3 days before hatching. So basically early in embryogenesis or late in embryogenesis. And so we expose the embryo to light, and then we look at how that affects their performance later after they've hatched and are doing these goal-directed behaviors. The results show that early embryonic light stimulation can modulate this particular behavioral lateralization, comparably to the late application of it via a different route. And so this kind of goes through this. So it is now well known that environmental light stimulation interplays with the genetic cascade of events in promoting brain specialization in 2 different classes of vertebrate, fish and birds. And so there are some reviews here. A complex chain of developmental steps leads to brain lateralization in zebrafish, starting with an asymmetrical expression of a gene network that controls the development of structural left-right differences within the epithalamus, including asymmetrical peripineal migration. So these are all involved in light perception and some other things in the brain. And so this exposure to light is affecting the ability of these chicks to basically respond to light later, and it affects their goal-directed behavior as after they've hatched. As a secondary consequence, in the transparent eggs of the zebrafish, an early action of light prompts functional brain asymmetries, including motor and sensory processing. Fries, which are baby fish hatched from eggs exposed to photic inputs during the first week after fertilization, prefer to attend to conspecifics with the left eye, whereas fries, whose embryonic development happened in darkness, do not display the same asymmetry. So again, you see this response to the environment and environmental cues based on how they were exposed to light as an embryo. And the eggs in these cases are very thin, so they let light in, or they don't let light, or you can have them reared in darkness. They'll let light in if they're exposed to light. And so that light actually kickstarts a complex set of interactions that leads to this asymmetry in the hatched individual and when they're performing goal-directed behavior. If light fails to reach the embryos in 2 distinct moments within the first week post-fertilization, the normal development of some lateralized behaviors is either compromised or prevented. For instance, darkness during the first day results in an inversion of the reaction to a dummy predator. After normal regimes, zebrafish avoid the predator appearing on the left side, whereas after darkness, they respond more intensely to a predator coming from the right side. In contrast, darkness during the third day prevents the appearance of any asymmetric response to the predator. So this goes back to this idea of critical periods. And so a critical period is a period in development where things are acquired in a very short time window. So sometimes we can think of this in terms of plasticity, or we can think of this in terms of just some mechanism that gets turned on at just the right time. And so if you think about this in terms of a developmental trajectory, we have developmental time here, and we have some trait that is going to emerge here. And so we have this time window here where there's this sort of discontinuity or this maybe phase transition between these two parts of development. And this is a very short time window we're talking about. So this is what we might call a critical period. So this critical period requires certain things to be in place, and this transition can occur. If things aren't in place, or if they don't have the right stimulus coming in, so you could have stimulus here, or you could have the right set of baseline conditions. It could be, it's probably, if you have something like, if you have a developmental mutant, for example, and they don't have a certain gene that's being produced, or they have a knockout of that gene altogether, that can affect the timing of this, the emergence of this trait. And so it can be outside of this critical period window, and you might end up with a developmental anomaly like this, where the phase transition doesn't occur normally, and you get an abnormality later on. So this would be like post-hatch. This would be hatching. And this would be the post-hatch phenotype. So the phenotype would differ based on what happens or doesn't happen during this critical period. So that's a very brief overview of critical periods, but I think it's essential to understanding what's going on here. So again, this is a nice paper that talks about some of these different effects of rearing the embryo in different conditions, either light or dark, and then what that does later on in the embryo's development. They talk a lot about asymmetries here, which is interesting because this is very far from C. elegans. This is in chicks, and they also talk about different they talk about fishes. So these are all vertebrate nervous systems. So it's a little bit different. There's that lateralization aspect that actually plays quite a role in some of these things. So this is there are a lot of interesting experiments you can do here. So one is here, reversing the eye exposed to light by untwisting the embryo's head and applying a patch to the right eye causes the pattern of asymmetries to be inverted. Note, however, that although light exerts such an important role in the establishment of lateralization, some forms of asymmetries, such as those associated with the unilateral eye used during sleep, or with the neural mechanisms of social recognition and imprinting, which is where a baby chick will imprint on a parent. This is a visual phenomena. Develop even if the incubation process takes place in darkness. So this is then the second paper is from behavioral brain research, and this is in zebrafish. So this is patterns of early embryonic light exposure determine behavioral asymmetries in zebrafish, the Habeniller hypothesis. So we talked a little bit about the pineal gland in chicks, and the pineal gland is sort of like a third eye in birds, and so they have this light sensor that's at one time when we had a common ancestor with hagfish. There is this third eye on the top of our heads. It was like a photic patch that would take in light stimulus. And in birds, because they have very thin they have a very thin skull, light comes through their skull and is processed by the pineal gland. And in our pineal gland, we don't get a light input, but it's usually used to regulate our moods, regulate some of our photic interactions, things like, when you get depressed in the wintertime, sometimes that's because there's a lack of light. and there's a pathway from the pineal through the pineal gland that affects that. So it's kind of an interesting organ. But this is actually the habenular hypothesis. So this abstract reads: releases of innate responses are more effective in many vertebrates when seen by the left eye. Again, we have this asymmetry between left and right eye. In zebrafish, Brachydoneorerio, this asymmetry is linked with neuroanatomical asymmetry of the habenular complex. So this is the habenular nuclei. This is an enlarged left lateral and right medial section of this nucleus. If habenular asymmetry is reversed, reader response to releasers shifts to the right eye. Light exposure, and they have a schedule here where it's they give it a certain treatment during the first few days of development post-fertilization, controls the patterning of this asymmetry. So this is actually very early in development. If you give it a certain amount of light exposure, just post-fertilization, you get this asymmetrical effect later on. We show here, using response to a model predator on day 7, that absence of light on day 1 PF, this is PF is post-fertilization. So there's actually a response to a model predator on day 7 of post-fertilization. The absence of light on day 1 post-fertilization alone causes high responsiveness to shift from left to right eye and to intensify. So there's a shift in the asymmetry depending on the stimuli that are provided to the egg, to the organism as it's in its embryogenic state. Absence of light on day 1 post-fertilization alone causes high responsiveness to shift from left eye to right eye and to intensify. Absence on day 2 post-fertilization or day 3 post-fertilization produces lesser shifts, whereas absence on all 3 days reduces responsiveness without any shift. So you can see that there are different shifts, different lateralities, and then if you don't have any sort of light stimulus at all, then there's no enhanced responsiveness at all. So we know that if there's no light input, that there's no enhanced responsiveness. This is due to light exposure of the in the embryo, and then there are these different asymmetries that emerge. A known disturbance of a gene, NRP1A, and its expression causes rerouting of the outflow of the left lateral habenula on the way station of the right medial habenula, providing a possible explanation of shifts. So this is a sort of an information processing thing that intersects with gene expression. So if you disturb gene expression of a certain gene, there's a different path that's taken by the information from one part of the habenula to another, and it causes this asymmetry to occur. And so we don't really know what maybe the selective pressure for that would be. Maybe there is a selective pressure. Maybe it's just neutral change that doesn't really, it just happens to be there and you get it triggered with light exposure. We don't really know. Variation in exposure of eggs to light is likely to produce inter-individual variation in the field. So again, this is variable by individual. And so this paper kind of goes through some of the evidence from chick, but then also integrates it with zebrafish. So again, this is a very interesting paper, and you see some of this evidence perhaps of critical periods here as well, because you need to do this during a certain time in development, a very narrow window, and these things have to be in place or else you can't get the effect. So if you expose it to light later in development, many days post-fertilization, you won't get the same effect. The final paper here of this group is early late embryonic stimulation suggests a second route via gene activation to cerebral lateralization in vertebrates. So this is all across vertebrates here. It includes zebrafishes and chicks, and probably some other organisms as well. I think they're actually looking specifically at chicks. But this kind of ties together the other two papers. This is in the journal Scientific Reports. In the abstract reads, genetic factors determine the asymmetrical position of vertebrate embryos, allowing asymmetrical environmental stimulation to shape cerebral lateralization. In birds, late light stimulation just before hatching on the right optic nerve triggers anatomical and functional cerebral asymmetries. So again, we have these asymmetries that we saw in the zebrafish and actually in the chick and the other experiments replicated here in another point in development. However, some brain asymmetries develop in absence of embryonic light stimulation. So brain asymmetries aren't just due to light stimulation. There are other reasons for them. But, this light stimulation, you can tie it to specific lateralizations in the brain in the post-hatch individual. And you can actually look at these critical periods or periods of time when the light has to be there for these things to emerge. Furthermore, early light action affects lateralization in the transparent zebrafish embryo before their visual system is functional. So this is important because I talked at the beginning of this section about how muscle twitching actually behaves like that too. Muscle twitching occurs quite commonly before muscle is connected to the connectome or the nervous system, the central nervous system. And so the same is also true that some of these lateralizations occur before the visual system is functional. So this is visual system independent. It's just something that happens in the embryo and then affects the visual system later. Here we investigated whether another pathway intervenes in establishing brain specialization. We exposed chicks' embryos to light before their visual system was formed. We observed that such early stimulation modulates cerebral lateralization in a comparable vein of late light stimulation on active retinal cells. So now they're looking at just stimulating the retinal cells without worrying about the visual pathways being in place or not. Our results show that in a higher vertebrate brain, a second route, likely affecting the genetic expression of photosensitive regions, acts before the development of a functional visual system. So in what they call higher vertebrates, there actually is a second route of light stimulation, and this is actually involves the pineal gland as well, pineal route of sensing light. This affects the genetic expression of photosensitive regions. So this actually is just kind of a secondary route that allows for genes to be expressed and to build these circuits and these pathways that are going to eventually become the nervous system. Now, at the end of the abstract, they say more than one sensitive period seems thus available to light stimulation to trigger brain lateralization. So they actually suggest that there are multiple sensitive periods or multiple critical periods. And we talked about critical periods, and I just showed you an example of one potential critical period. But you can have critical periods throughout development, and they can build upon one another. So you can have one critical period that has to be, that allows for maybe some change to occur or some stimulation, and then another critical period later, and then another one later, and they compound upon one another. So if one critical period window is missed, the next critical period window might be met. The criterion for triggering the stimulation might be met there, but because it missed that first window, the effect might not be as great. So you can see how that works, where you have these almost like these steps in development that are enabled by critical period windows, where you get some critical period where there's some sort of rapid acquisition of something. It could be information for gene expression. It could be some sort of other environmental conditions, and then that translates into some sort of that maybe translates into normal growth or some sort of plasticity that can affect the output or the post-hatch embryo. So this is very interesting work. I think it fits well into some of this other stuff. So this is a chick, this is their asymmetry that they're exhibiting here in the behavioral world post-hatch. And this is, of course, after they've been exposed to these things in the embryo. There are other things that they've done. So they've done some experiments here. They're looking at how chickens are pecking, and they're looking for these asymmetries in behavior. And so these are all set up in the embryo based on the amount of light that they're getting. So that's an interesting new area, or I don't know if it's a new area, but it's definitely something that we might follow up on later. So this next one is a collection of papers, and it's on new forms of morphogenesis. And so it's kind of they call it pushing, pulling, and non-reciprocity. And so there are a number of things going on here that I want to kind of go over. And so this first paper is from Quantum Magazine. It's an article that basically sets this up as an alternative to the reaction diffusion models of Turing. So embryo cells set patterns for growth by pushing and pulling. So patterns that guide the development of feathers and other features can be set by mechanical forces in the embryo, not just by gradients of chemicals. And so the reaction diffusion model of Turing really relies on chemical gradients. They rely on differences in chemical gradients and other types of chemical interactions to form patterns. But in this case, what they're arguing is that, and you'll see that there's a group of people working on this, at least one group, where mechanical forces are really driving a lot of this pattern formation. We've talked about mechanical forces before being very important in embryogenesis, in driving some of these processes where you get like convergent extension and some other stages in development where the embryo changes shape from a sphere to something asymmetrical. And so, but they're also arguing that mechanical forces play a critical role in pattern formation. And so one of the longest standing questions in biology is how a living thing that starts as an embryonic blob of uniform cells morphs over time into an organism with diverse tissues, each with its own unique pattern and characteristics. And so this talks about the model, Turing's reaction diffusion model, which has had many successes. But a growing number of scientists suspect that Turing's theory is only part of a story, the story of embryogenesis or morphogenesis. In my opinion, we've been blinded by how idly it should be applied simply because of its beauty, says Amy Shire, developmental biologist at Rockefeller University. So she views physical forces of contraction and compression being also very central to this pattern formation story. So I'm going to go, this paper here in Cell, I'm going to go over that paper. And Alan Rodriguez is their co-author on this. And they show that mechanical forces could induce embryonic chicken skin to create follicles for growing feathers. So you can actually use mechanical forces to induce follicular growth and thus feather formation. And this is something we typically thought of as a chemical problem before. So, it's like there's a chemical gradient, there's a distribution of feathers, and, it depends on how that chemical gradient is shaped, that determines the shape of the feathers on the phenotype. In this case, they're saying that you can have physical forces that act in a very similar manner. So just as surface tension can pull water into spherical beads on a glass surface, so too can the physical tensions within an embryo set up patterns that guide growth and gene activity in developing tissues. So as the cells in the tissues push and pull on each other and on the support of protein scaffolding, or the extracellular matrix to which they are linked. And so some researchers have suspected that these forces, coupled with changes in pressure and rigidity of the cells, might direct the formation of complicated patterns. But we've talked about this before, where the cells can change their shape, and they can do a lot of interesting things, especially collectively. But in this case, we're actually suggesting that this is a key part of pattern formation. And, it may be that there's some sort of lawful behavior here. We don't really know. Until now, though, no studies were able to tease apart the effect of these physical forces from the chemical stew in which they simmer. And so they did these experiments in chick embryos. So again, we're back to the chick embryos. So they were able to take some chick embryo cells, skin cells, and grow them in culture. They watched as the skin cells self-organized into a ring on the floor of the dish, like a 2D version of the ball of cells that the embryo normally becomes. The cells pulled in collagen fibers in the extracellular matrix as they assembled around themselves. And then over 48 hours, the fibers rotated, bunched together, and then pushed each other apart, forming bunches of cells that would become feather follicles. So they're basically forming these clusters from just basically a distribution, random distribution of fibers, and they're forming these patterns. So this is a lot like Turing pattern formation, where you get these, sort of this homogeneous gradient or this randomized gradient, and then you end up with these patterns, these stripes or spots or whatever. In this case, they would basically be spots. So you can see in the image here that form the basis for these feather follicles. Okay, so this is such a clean, simple experimental setup where you can see a beautiful pattern come out and quantitatively control it. And so you can do a lot of things. This is a very quantitatively oriented experiment. So, there are a lot of parameters that you can adjust and look at like how this works. So that's nice. So in this paper, Bending Gradients, How the Intestinal Cell Gets Its Home, they go over the summary. We address the mechanism by which adult intestinal stem cells become localized to the base of each villus during embryonic development. We find that early in gut development, proliferating progenitors expressing ISC markers, which are intestinal stem cell markers, so they have molecular markers that they can identify progenitor cells as possessing, are evenly distributed throughout the epithelium in both chick and mouse. So they have an even distribution across that epithelial layer. However, as the villi form, the putative stem cells become restricted to the base of the villi. This shift in the localization is driven by mechanically influenced reciprocal signaling between the epithelium and the underlying mesenchyme. So as we saw in the graphical abstract, we can see these two layers, and there's reciprocal signaling. You can see the arrows here between Wnt and SHH and BMP. And so there's the signaling, both positive and negative signaling. So, and then we have buckling forces physically distort the shape of the morphogenic field, causing local maxima of epithelial signals, in particular SHH at the tip of each villus. This induces a suite of high threshold response genes in the underlying mesenchyme to form a signaling center called the villus cluster. Villus cluster signals, notably BMP4, feed back on the overlying epithelium to ultimately restrict the stem cells to the base of each villus. So it's this combination of signaling and then buckling forces through sort of these bucklings of the initial bucklings of the tissue that drive the formation of these villi. So it's basically driving the positive feedback, I suppose, that drives this process forward and refines these villi from mere buckles to these formed villi, and it goes across the surface of the intestine. So they show some images here. They show an example here where you have these buckles that form villi eventually. It's kind of like they look like fingers coming out of the surface of the intestine. You can see as they're refined. And then at hatch, you have these fingers that come out, these villi. And so that, now I'm going to go back to the original paper. I said that I was not going to present that, but I actually, now that we've established what's going on, then we can actually go to this paper, which is the paper that was featured in the popular article. And so this paper is titled "Reciprocal Cell ECM Dynamics Generate Supracellular Fluidity Underlying Spontaneous Follicle Patterning." So again, this is the graphical abstract. This is out of Cell. So Cell likes to do graphical abstracts. We have the organ here where you have two layers, and you have this uniform embryonic skin, and you have some symmetry breaking event where you get this buckling and then this refinement of the buckles, and this is the follicle pattern. So this is a little bit different This is follicle patterning. You get they're able to do this in culture. Then they're able to show these orderings. So as the cells get plated on this two-dimensional culture, the cells form a ring. And then this ring becomes ordered over time. So these are isotropic cells. These are just kind of laid down in a predictable pattern around in a circle. Then they become aligned with the pattern axis. Then they become ordered as an active fluid. And then you get these aggregates that form from this ordering. So they become aligned, they become ordered, and then they become periodic. We talked about this in the liquid crystal biology lectures, and this was about maybe 6 months to a year ago, where we talked about nematic orderings. And so this is very similar. This is an active fluid. This is a soft material type approach to this. So you get the soft material alignment. These are collective behaviors among cells, and they have these different properties. So refer back to those lectures, or the D-GNN book on active matter, soft matter. Then in cells, you see that you have this extracellular matrix here. You have contractile cells that align the extracellular matrix. This extracellular matrix increases its contractibility through calcium signaling and calcium ions, and then you have this contractile instability. So the highlights of this article are: an ex vivo assay reconstitutes the process of embryonic skin symmetry breaking. Mesenchymal mechanics are sufficient to spontaneously generate morphological pattern. Contractility-dependent cell extracellular matrix interplay, so the interplay between the cells and this extracellular matrix down at the bottom here, creates supracellular structure. So supracellular structure is the sort of how in culture you can have this alignment and then this clustering. And this is just in culture, but in a tissue where you would have this extracellular matrix, you actually get these clusters and they form with the extracellular matrix around them. So you get these instabilities, you get these dynamics that you don't necessarily get in cell culture, although you can demonstrate the pattern formation in cell culture. Theory and experiment show that cell ECM layer behaves as an active contractile fluid. So again, this is the contractile fluid here. You have this active fluid and has this sort of these properties of soft active materials. So why don't we go over the summary? During vertebrate embryogenesis, cell collectives engage in coordinated behavior to form tissue structures of increasing complexity. And this set of experiments is in avian skin. So in avian skin, assembling into follicles depends on intrinsic mechanical forces of the dermis, but how cell mechanics initiate pattern formation is not known. Here we could reconstitute initiation of follicle patterning ex vivo, which is in the cell cultures, using only freshly dissociated avian dermal cells and collagen. So they're plating this down, they're watching this formation, and then they're looking at the interaction between cells and extracellular matrix separately. And they want to see, this pattern formation, if it can be done in a dissociated context, but also in a context of a tissue. So we find that contractile cells physically rearrange the extracellular matrix, and that this matrix rearrangement further aligns cells. So the extracellular matrix reinforces a lot of these pattern formation trends. This exchange transfers a mechanically unlinked collective of dermal cells into a continuum with coherent long-range order. So this is this nematic ordering where it's long-range, and it isn't just neighboring cells, it's, the 50th cell down the line. It's also ordered in the same way. So you have to, that sort of, we don't want to call it global ordering, but that sort of long-range ordering is essential for this sort of pattern formation. Combining theory with experiment, we show that this ordered cell ECM layer behaves as an active contractile fluid that spontaneously forms regular patterns. Our study illustrates a role for mesenchymal dynamics and generating cell-level ordering and a tissue-level patterning through a fluid instability, processes that may be at play across morphological symmetry breaking contexts. And we talked a lot about symmetry breaking and instabilities and things like that in our group as well. So, we go back to some of the lectures on that if you want to know more. So I don't know if there are any nice figures in here. This just sort of shows the alignment in their experiments and then pattern formation. So it's not just enough to have pattern formation, you have this alignment of cells that has to initiate pattern formation. So you can see that here. And then, this is the protocol for what they're doing. It's worth noting, though, that you have some of these things. So let's take this figure here. You have figure 1A. This is a bright field image of day 6.5 embryo backskin. So this is from the embryo. And this is at day 6.5. And then F is a bright field image at day 7 embryo backskin. So this is half a day later in development, E7. And so so this just kind of shows the patterning as it's happening. So it aligns first in that E6, E6.5 window, and then E7, you start to get these patterns, these clusters. So this whole process unfolds pretty quickly. And of course, we talked about critical periods. And so there's a critical period here, I suppose, between E6 and E7, where a lot of this has to unfold. And of course, if there are defects in the embryo, if there are developmental mutations, or if there are environmental issues that are going on with both the extracellular matrix or something happening in the cells, what they're expressing, where they are mechanically, then this doesn't happen, or it happens in a different manner, and it doesn't necessarily form the right pattern. So this is, this kind of goes back to our critical periods discussion here. And so this just kind of shows in culture how this forms. You get these rings, and then you get alignment of the cells, and then you get pattern formation. Okay, so that's I think all I'm going to talk about with that paper. So we have two examples of that. Now I want to talk more directly about living crystals and some of the liquid crystal biology stuff that I talked about earlier. If you go back to DeVoWorm, it talks about soft active materials and some of these phenomena in crystals, but then makes the application also to some forms of single cell biology. And of course, this has been worked out in more recent past in biophysics, in the biophysics literature. Now, this paper is interesting because it talks about living chiral crystals. And so it talks about the odd dynamics of living chiral crystals. And so I think you've seen with the last set of papers that we're dealing with a set of similar issues. We're dealing with pattern formation, we're dealing with physics, we're dealing with some of these things that need to be in place for pattern formation. And it's not necessarily chemical interactions. But so this paper talks about living chiral crystals. So the abstract here reads, active crystals are highly ordered structures that emerge from the self-organization of motile objects, which means they're moving objects, and have widely been studied in synthetic and bacterial active matter. So this is, synthetic active matter, things like, collective surfactants and things like that. In bacterial active matter, of course, are bacterial colonies that form patterns around stimuli in the local environment or other types of, colony formation. Whether persistent crystalline order can emerge in groups of autonomously developed multicellular organisms is currently unknown. But here we show that swimming starfish embryos spontaneously assemble into chiral crystals that span thousands of spinning organisms and persist for tens of hours. So we're looking at starfish embryos here. And I think, okay, this is the paper from Peter Foster. So Susan earlier mentioned Peter Foster, and this is that work of that group. So this is swimming starfish embryos, and they're spontaneously assembling into these crystals. Combining experiments, theory, and simulations, we demonstrate that the formation, dynamics, and dissolution of these living crystals are controlled by the hydrodynamic properties and natural development of embryos. Remarkably, living chiral crystals exhibit self-sustained chiral oscillations, as well as unconventional deformation responses behaviors recently predicted for autoelastic materials. Our results provide direct experimental evidence for how non-reciprocal interactions between autonomous multicellular components may facilitate non-equilibrium phases of chiral active matter. So this is, again, this is now with a very different type of organism, starfish embryos, and we have symmetry breaking. We have some of these active matter issues, and so this is a particular interesting class of non-equilibrium symmetry breaking phenomena comprises the active crystallization processes recently observed in colloidal and bacterial systems. So colloidal systems are systems of particles, usually materials like sand or other types of soil or other types of things that have this, collective behavior, and they form states of matter. Unlike conventional passive crystals, which form on lowering the temperature and often require attractive forces, active crystallization arises from the particle's self-propulsion and can occur even in purely repulsive dilute systems. A long-standing related unanswered question is whether groups of multicellular organisms can self-organize into states of crystalline order. So crystalline order is this sort of macro state. So in this case, you have starfish embryos. They're independent, but then they form these macro states. And these macro states are the crystals that you talk about here. So they're not, I guess they're living systems, but they're not like, the macro state isn't necessarily a living system, but the constituent parts are. So here we report the observation of spontaneous crystallization in large assemblies of developing starfish embryos, Patria miniata. Our experimental observations show how, over the course of their natural development, thousands of swimming embryos come together to form living chiral crystal structures that persist for many hours. And they have a video of this, which I'm not going to show, but it's a supplemental video to this paper. In contrast to externally actuated colloidal systems, the self-assembly, dynamics, and dissolution of these LCCs are controlled entirely by the embryo's internal developmental program. So this is where the embryos at a certain life history point in their developmental history, they form these constructs and then they dissolve as quickly as they formed. So this goes back also to the critical periods, because you have these little periods where they form these different structures. Once formed, these LCCs exhibit striking collective dynamics, consistent with predictions from a recently proposed theory of audioelasticity. And so audioelasticity is, well, we'll talk about that in a little bit. I'd like to go through these images, actually. So these are images of this crystal formation. These are the embryos here, and they're forming this crystal structure. And then they're forming patterns. So you can see that the embryos aggregate and they form these crystal structures. And so they self-organize into living chiral crystals. Time sequence is a still images showing crystal assembly and dissolution. This is A is at 2 hours, and then this one's at 5 hours, 26 hours, 35 hours, and 38 hours. Basically, it shows a sequence where they start to build up, they build this crystal. You can see pattern formation within the crystal, and then it dissolves. And so you get the dissolution at 38 hours where they break apart. This C actually is, so the embryo morphology and flow fields, which are here, change with developmental time. So you can see over developmental time, at fertilization, you get this, this basic polarity. Then at 12 hours, you start to get this internal structure in the embryo, 26, 38, and 44 hours. So 38 hours, when it's dissolving, it looks much different than what it looks like at 12 hours or 0 hours, but actually not that much different than 26 hours. It's just that the dynamics change, the flow field dynamics change. So, and then this C actually is embryos assembled in a crystal perform a global collective rotation. So once they get into these crystals, they form rotations, and they get through the water column. And I don't know if they talk about exactly about why they do this, but I think this is just characterizing this more than anything. So this is very similar to Volvox colonies and what Volvox colonies do, which is that they form these larger structures, and they use this to navigate their environment. And they do this when they're swimming near rigid surfaces or other places in the water column. And so single embryo properties facilitate formation, rotations, and dissolution of clusters. So single embryo properties, as they grow, they sort of govern this collective. If they're synchronized in their in their developmental period, then this drives this process. And even if they're not necessarily synchronized, you still get like the dissolution of crystals as some of the embryos age out. So this is an example here. Embryo spinning frequency, and then this is from 0 minutes to 20 to 40 to 50 to 60 to 200 minutes. And you can see that in this case, the crystal is persisting over this time period. And so, yeah. I don't know if they talk about, so the temporal evolution of crystalline order. So a striking feature of the LCCs is that they nucleate, grow, and dissolve naturally as embryos progressively develop. To quantify the evolution of crystalline order, they calculate a local order parameter, and then measurements determine the local phase representing the crystal orientation, as well as the magnitude of hexagonal order. Initially, small clusters merge together along different crystal axes, resulting in grain boundaries and broad distributions of these parameters. Within 5 hours of crystal formation, LCCs undergo rapid internal restructuring during which subdomains align. This results in nearly defect-free crystals with a high degree of hexagonal order and a narrow distribution of local bond orientation. This highly ordered state persists for several hours. And then as development progresses, you get changes in morphology in the surrounding flow fields, and that kind of breaks up the crystalline order. So these flow fields change as the colony sort of forms these crystals, and they kind of rotate around. And so this is all kind of almost like a self-contained system, and it's driving its own dynamics, and the constituents are driving the dynamics. It's really kind of a fascinating system. So these actually show defect streams and displacement waves exhibit signatures of autoelasticity. So this is what they talk about, autoelasticity. And I'm trying to find, okay, here's maybe a definition of autoelasticity. So starfish embryos are inherently chiral, which means they spin in a certain direction, or they have a handedness, so they have like a bias towards one direction or another. They spin about their AP axis, which is anterior posterior axis, in a left-handed manner. So they spin left-handed, and they prefer that orientation. This chiral spinning motion leads to distance-dependent transverse lubrication interactions between pairs of embryos. And so they want to look for autoelasticity. So autoelasticity can emerge in active isotropic solids that are chiral. Autoelasticity theory complements a conventional elastic response of passive isotropic solids to compression and shear. And so to identify signatures of autoelasticity in our experimental data, we use the fact that LCCs typically harbor lattice defects. These defects locally deform LCCs with the deformation field encoding about the effective material properties. We quantify deformations in the frame co-rotating with the cluster by measuring the displacement of embryos at position r and time t for regular lattice. And so, it's basically autoelasticity or the odd moduli as well. They have this some sort of defect in the colony. And so this is what they're referring to. The presence of odd moduli, which are a derivative sort of autoelasticity, raises the possibility that LCCs can support self-sustained chiral waves and strain cycles, similar to those recently predicted in autoelastic materials. Okay, so you have these elastic. The oddness, I guess, comes from some sort of asymmetry or fluctuation in the material. So this basically leads them to some sort of theory about this, and they can talk about this in terms of these sort of living chiral crystals and liquid crystals and soft active materials. And so I think this ties together a lot of this sort of these new directions in morphogenesis, and I think it provides a nice alternative to the Turing reaction diffusion work. Okay, thanks for joining me. All right, well, thanks for the updates again, and if you want to, we'll meet this coming Monday again, back to the regular schedule, so if people want to present anything then, they can. If not, we'll have some things going on. I'm sure people will have updates for things. Okay. All right, thank you. Have a good week.
Bye.
Bye.
Thank you. Bye.
Bye.